Yes. Oh, and so I just want to remind you before we get further in that you should type your questions into the question and answer box, not into the chat, and you'll see that along the bottom. It's the Q&A. And at the end of the seminar, Erica Goss and I will take turns reading off all your questions. So now I'd like to move on to our seminar. So I'm very excited that Jared LeBoldus is here today to tell us about an interesting paper that he had in our February issue of MPMI, looking at using population genomics as an entry point to understand something about how plants and microbes interact. And so we're all fascinated by the intimate connections between plants and microbes. And there are many, many different ways to study this. And what Jared's going to be telling us about is a way to actually use the population genomics of the host plant itself to understand something about how humans have interacted with the interaction between a plant and a tree pathogen. So the populace and a tree pathogen. So very interesting population genomics, an area that we're really trying to expand at MPMI. We've had a few interesting papers in there, and I particularly enjoyed this one, and that was why I asked Jared to tell us about it. And I mentioned that 2020 has been a difficult year, as I'm sure you all know. And in addition to pandemic, economic distress, lots of other things going on, we also have many natural disasters. And one of the natural disasters facing us right now in the United States is a series of wildfires, many wildfires all over the western part of our country. And I'm very glad that Jared has been able to join us today, although he is not joining us from his office because of all the wildfires around him in Oregon. So hopefully things are getting better. But welcome, Jared. And I'd like you to start by just telling people just a word or two about who you are. And then just a reminder that this talk is about a paper that he recently published in MPMI. And you can go to that paper if you want to dive a little more deeply into any of the data, the methods, et cetera. And if you want to make the rest of us go away and just focus on the speaker, you can just click speaker view at the top. Thank you, Jared.
Thank you very much for the introduction and for the invitation to speak today. I'm excited to be able to talk about this paper. It was a fair bit of work to get the population collected and do the sequencing and then the analysis afterwards. And so I'm really excited to talk about it, and hopefully we'll have lots of good questions and discussion afterwards. So just to introduce myself, I am a assistant professor at Oregon State University. I'm a forest pathologist, and I work on kind of two worlds. The first world is I study host-parasite interactions in poplar, and with the pathogen I'm going to talk about today, Spirulina muciva. And we're basically looking to identify genes that mediate that interaction. And that's what kind of led us to this population genomics approach that I'm going to talk about here. The other hat I wear is a forest pathologist who works out in southwestern Oregon in Curry County, where there's an outbreak of the invasive oomycete pathogen Phytophthora ramorum. So in that case, I try to help management, so I kind of wear two hats. But today I'm happy to be here and talk about poplar, which is something I've worked on since my graduate career, I guess 15 years ago now. So today I'm going to talk about population genomics reveals anthropogenic mediated dispersal of a tree pathogen. Okay. And I always like to start these talks by thanking all the people involved, mostly because sometimes I don't want to get cut off, and I know that's not an issue with this talk today, but I really like to highlight the roles that people play in the whole research endeavor. And without all these people, and all these collaborators, none of this would really be possible. So first and foremost, that picture is my lab group. So I'd like to thank all of them for all the work they do, and without them, none of this would be possible. I'd like to thank our collaborators at the University of British Columbia. So this is Richard Hemelin, Nicolas Faux, and Monique Sakalidis. And they basically helped us in terms of providing a large portion of the isolates that we resequenced, and they helped us with the development of the paper, and they're co-authors on this paper. I'd also like to thank collaborators at the Department of Energy and Oak Ridge, who've helped us a lot with the poplar side of these things. And then of course the funding agencies, so USDA, NEFA, the Department of Energy, and OSU as well. So thank you for all your help. Okay, so now what I'd like to talk about a little bit is the fungal pathogen. So Spirulina muciva, which used to be called Septoria muciva, is an ascomycete. It's native to the eastern United States, and it causes two characteristic types of diseases. So one is Septoria leaf spot, and the second is Septoria stem canker. This disease, or this pathogen, or this organism was first described in 1884 by Peck, and it was described infecting Populus deltoides in New York State. And the symptom that it caused were these leaf spots that you see on the left side of the screen here. The other symptom and disease it's called is a stem canker. And it's really the stem canker that started drawing attention to this disease. It causes severe problems on poplar plantations where they're used for biomass and bioenergy, and basically it can have a big impact, wiping out an entire plantation in just a couple of years. So you can see here, there's two pictures. There's the broken stem of balsam poplar that's been infected by this pathogen, and here you see the lesion of a stem that's been inoculated in a greenhouse inoculation, which I'll describe in more detail later. So to give you an idea of the impact of this disease, so I mentioned earlier, poplar are typically used for biomass and bioenergy, or at least that's the idea behind poplar plantations. And really, you know, the area that's been planted to these has kind of expanded across the United States and Canada over time. It began in the eastern US and has kind of spread westward since then. And I think that's important to keep in mind, and we'll come back to that later on when we talk about the population dynamics of this pathogen. So this is just to give an idea of some of the impact that this disease has. So one plantation in Michigan, 5 years after planting, 86% of the trees had Septoria cankers, and 2 years later, 69% had broken tops. So these plantations need to be in the ground for about 20 years in order for them to be productive and return on the investment. And as you can see, if most of the trees are dead within 7 years, that's not really going to work. Another example, you know, in a trial with 56 different genotypes of balsam poplar, at least one had stem cankers 7 years after they'd been planted. So it's a significant problem. The other thing that we've started to notice that as this poplar production moves westward is this pathogen has seemed to move along with it. And you know, most recently in 2007, and now actually in 2020, we've identified this pathogen in both the Western United States and Western Canada. And I'm going to come back and talk about some of the new work at the very end, just a couple slides about this. And really the thing to keep in mind here is that most of the really desirable poplar trees, the hybrids that are planted in the north central and eastern United States are susceptible to this disease. And so the real focus has been trying to find resistance and trying to basically limit the spread of this pathogen. So the objectives of this talk, or what I want to talk about today, and this paper, were basically to get an idea of the population structure of the pathogen in North America. We want to characterize the connectivity among populations. So this is basically to give an idea of how this pathogen might be spreading across North America. We were interested in identifying genomic regions with signals of local adaptation. And the idea here being that if we could find genes or groups of genes that might demonstrate differences between these, they might be targets for future downstream functional work. And then finally, I'm going to discuss a little bit the recent introduction into Oregon and some of the work that we've done around that. Okay, so Spirulina musiva, the population that we resequenced for this paper, there's about 122 isolates of the pathogen, and they were collected from basically across North America. There were some holes in the distribution, but I think in general, we did a pretty good job capturing that. And that represented a lot of work for over years of different people who had spent time collecting isolates of this disease. They were collected from a variety of hosts, variety of symptotypes, and a variety of ecosystems. So we collected them both from natural ecosystems and from plantations. And then once we had kind of accumulated all these isolates, we grew them in liquid culture, and then we extracted DNA. And then they were each sequenced on a HiSeq 3000. And the sequencing was 150 base pair paired end reads with a median insert size of about 500 base pairs. And then once we got the results from this, they were mapped to the reference genome of Spirulina musiva, which is available on the JGI website. So just to kind of summarize the results from the resequencing effort. So there were almost 900 million reads that were sequenced. And each of the isolates or individuals had about the depth of coverage range from about 17 to 80x. And then once we had all this data, the next step was to filter it. And so I have highlighted here on the right-hand corner, Dr. Javier Tabima, who is the first author on this paper and basically handled the majority of the bioinformatics work and the population genomic analysis. So we set up a filtering pipeline where basically anything with any position with a depth less than 4x was removed. Anything with a mapping quality less than 44 was removed. Positions, so these are individual positions in the genome. If they had greater than 40% missing data, they were removed. And then the minimum allele frequency cutoff that was used was less than 0.014. And so basically what that is that represents that fewer than 2 individuals out of that full 122 that we resequenced would have not had that allele. So when all this filtering was done, and I'd be happy to discuss that in more detail afterwards, we ended up with 120,000 approximately SNPs that were spaced across the genome and across the 13 chromosomes of this organism. Okay, and so that basically led us into some of the analysis that we were allowed to conduct, to basically try and understand the population dynamics of this organism. So here you see two different figures from the paper. And before I go into discussing those a little bit, I just wanted to briefly bring up some basic descriptive statistics. So basically across this population, we saw low to moderate levels of genetic differentiation. So the way we measured genetic differentiation was through FSTs, and we did pairwise comparisons between the different provinces and states where these isolates were collected. So here you can see a heat map, and you have the pairwise comparisons and that FST value on this heat map. And as you can see, they kind of range between 0.01 to all the way to about 0.33. So anything greater than 0.2 can be considered relatively differentiated, whereas the smaller the number becomes, the more similar those populations are in terms of their genetic diversity. So the next thing that we did was basically construct this phylogenetic tree, or this maximum likelihood tree. And that let us look at the distribution of these individuals and how related they were to each other in terms of their genetic diversity. And what this analysis basically shows is that you have 2 large groups that are differentiated from each other. So you have this upper group, which is basically a population from the Eastern US and Canada. And then we have this lower group that is the Southeastern United States. And then within this larger Canadian or US Canadian population, we found this subgroup or subclade, which is the BC2 population. And I'm going to come back to this later because this becomes interesting in terms of how different it is and how it's related to some of the stuff we found more recently in Oregon. So then when you have we look at the second part of the figure. So this is a maximum likelihood tree. And then in the second part of the figure here, you see this is a DAPC. So it's a discriminant analysis of principal components. And basically what this does is group each isolate or each individual into groups based on their similarity to all the others. So as you can see here, what you want to do is look at the colors. So you have this BC2 group, which basically is all the same color. And then you have, you'll notice this in the Canada US group, you have this green, the red, this kind of bluish green. Basically, similar colors grouped together from these similar areas. So this DAPC basically supported our phylogenetic analysis. So we were then kind of interested in trying to determine how, we have these two big groups and this subgroup within it. And there's a way to look at the genetic diversity and among these individuals and determine what's the best number of clusters or number of groups. And so that's what you see here on the left side of the screen. And so basically what we did was we used a clustering approach that let us test without any prior information, a whole bunch of different clusters. So 0, 1, 2, 3, 4 clusters. And the best fit for this turns out to be 3 clusters. And so when we go back to the previous slide where we had the US-Canada population, and then we had the southeastern US population, and then we had this BC2 cluster, it turns out that this BC2 cluster is different enough from the other two that it basically breaks out by itself. And that's really interesting when we start going and looking at the geographic distribution of this pathogen. So the second figure you see on the slide here is a map of North America. So you have Canada up here, and you have the southeastern US here, and then the rest of the United States over here. And basically, what I want to point out here before I talk about some of the statistics are that so there's this province here, British Columbia, and there are 2 separate groups of individuals from British Columbia. So we have one that's found on the coast that was introduced from the coast, and a second group that was introduced in the interior. So this interior group is this BC2 cluster that you see in purple above up here. And basically it's geographically separated from the other population and looks like it was a different route of introduction. Looks like it came from somewhere else. So that brings up this whole idea of gene flow and the movement of individuals across this landscape. So how did this happen? So I mentioned earlier on in the talk that this pathogen is native to the eastern United States and Canada, and basically seemed to start showing up as poplar trees got planted further and further and further west in the United States and in Canada. And so what we were able to do with this population after we kind of described the structure was to look at migration or estimate migration. And so we have this inset figure here above where you have these 3 populations, so BC2, US, Canada, and the southeastern US. And we were able to estimate migration or mutation-corrected migration rates between these different populations. And so basically these are the M values. And the bigger the M value, the more migrants or more gene flow that there is estimated to be occurring between these different populations. And so I think the things to highlight here are the fact that the southeastern US appears to be a big source for the population, or the BC2 introduction. And most of the other gene flow seems to be coming out of the US-Canada population. So that's this big kind of teal color that occupies the center of the continent. And there's movement back and forth between them. But most of that movement seems to be coming out of the US-Canada subpopulation. So another thing we were interested in looking at with this was to figure out the extent of sexual recombination or admixture that was happening between these different populations. So we were interested in basically determining if there looked like there was lots of sexual recombination or if there was limited sexual recombination, and then how these different populations differed from each other. And the reason we were interested in looking at this is that if you have limited sexual recombination, it can be kind of a clue that highlights a recent introduction of a pathogen. Whereas older, more well-established populations have a more extensive amount of sexual recombination that occurs as long as you have the mating type system of the fungus allows that to happen. So this first figure we see on the left-hand side is basically an estimate of linkage disequilibrium. So this tells us how fast or across how many base pairs LD decays. And as you can see, when it drops, basically when it drops below this 0.2 threshold, that's what we're interested in looking at. And as you can see, the Southeast US population and the US-Canada population both have rapid drops in linkage disequilibrium, which suggests there's a fair amount of recombination going on, and these populations have probably been established for a longer period of time. And then you have this BC2, where you have a slower rate of decay of linkage disequilibrium. Now, there's one important caveat I have to throw in here, is that when we compare these three lines to each other, the population or sample sizes for the blue and green line are a lot larger than they are for the BC2 line, just because we had more individuals from those populations. So that could also be a reason why you have these differences in the decay of LD. The second figure that I want to show you here is basically a simulation. And the idea here is that what we can do is compare the index of association, so how tightly linked two different positions are in the genome of an organism, to a sort of simulated situation. So here we have D, E, F, and FG are indicated here. That's BC2, Southeastern US, and the US-Canada population. And basically what we did was estimate the index of association for these different populations. And we compared those to scenarios where the SNPs or markers were totally unlinked, were 25% linked, 50% linked, 75% linked, and 100% linked. The idea being here that this would be 0% linkage would represent panmixia, so everything is freely interbreeding. And then you'd have this 100% linkage, where basically that would be a clonal population or a clone of that individual. And so really, it's interesting to note here that we really have evidence here for a significant amount of recombination or sexual recombination in these different populations. Now you'll note that the Southeastern US and US-Canada both seem to have more of this than BC2. The letters, of course, on this represent significant differences. And this is interesting from a sort of mycology standpoint, because when you look through the literature, you really see no evidence of the sexual structures of this pathogen. No, they've been described, but people don't seem to find them in the field. And so we really thought that most of the reproduction of this organism was asexual, but that doesn't really seem to be the case when we look at this simulation. Okay, so I've kind of hinted at the fact that the movement of this pathogen seems to be linked to the movement of poplar across the United States and Canada. And so I'm not sure how many in this audience are familiar with how poplar are propagated. So basically what you do is you take a 10-centimeter piece of wood that's the current season's growth. So you see a big pile of sticks there. and you stick those in the dirt, and 3 months later, you'll have a tree that's about 2 or 3 feet tall. So that is ideal for working and studying with poplar compared to any other woody plant. It's a real advantage. They're easy to clonally propagate. But that also makes it really easy to take cuttings and move them across North America to wherever you want to have them to allow to create one of these plantations. And it turns out that really, you know, in the early 1900s all the way up to the 1990s, these sticks were kind of freely exchanged between the United States and Canada and between breeding programs. And we hypothesized that this is sort of the likely route that this pathogen kind of made its way across North America. And so I think the thing here that's really interesting is, and this is a little bit of a side and not in the paper, but I had a postdoc in my lab who was interested in trying to figure this out. And so she took isolates of this fungus, inoculated some trees, and then did serial sections through the lenticels, which are the way we think this pathogen gets in. And it turns out that you can see mycelia, conidia, and hyphae inside these lenticel structures, so those white lines you see there, without any symptoms actually being present. So we think that in certain cases, there's asymptomatic colonization of these lenticels that's allowing the movement of, or sort of facilitating the movement of the pathogen along with the plant material. Okay, so whenever you have a genomics analysis, the question always comes up, okay, you might see these differences in these populations, but what does that really translate to in terms of disease? Do we see differences in terms of phenotypes that are caused by this pathogen? So I'm going to sort of describe a really large scale experiment. So I had two postdocs in my lab who basically undertook doing these inoculations. So we took 5 genotypes of Populus trichocarpa that we had previously phenotyped. So we knew some were resistant, some were intermediate, and some were susceptible. And then we inoculated all those trees. And then we scored the phenotypes. And so we did 2 different types of scoring. One was just the lesions per centimeter. And here you can see basically on each of those white lenticels, you have a black lesion developing. And the second was more of a disease severity score where we rated the level of disease on a 1 to 5 scale. And so you can imagine we had 122 isolates, 5 genotypes, and this was replicated 5 times. So that was a lot of trees and a lot of work and a lot of inoculum. And I really want to give a shout out to these 2 for facilitating all that greenhouse and growth chamber work. Okay, so when we start to group these 2 phenotypes by the genetic clusters, we start to see something really interesting. And I don't want to spend too much time on this, but I really want to just draw your attention to the fact that just like in terms of the genetic diversity of the southeastern US and the US-Canada population, the phenotypic diversity is fairly similar to each other. And in terms of the variation is also fairly similar. And then we see that when we look at this BC2 cluster, we don't see the same pattern. So it seems to be a lot less aggressive or a lot less virulent. And so this was really, really exciting. And what this sort of gave us the hope is, okay, if there's these big differences we can see, maybe we can take we can try and do some association mapping approaches, or we can do some other selective sweeps in the pathogen population to try and identify candidate genes that might be causing these differences. And so we did a fair amount, or Javier did a fair amount of work on this. We've had some success with the association mapping, which I don't want to scoop us by going into that right now. But we also have had some effect just looking at predicted genes and differences in the abundance and frequency of those genes in these different populations. And I'm going to come back to that at the very end. But before I do that, I want to talk about something that we recently found in Oregon. So in Eastern Oregon, as part of a Department of Energy growth and yield trial, we found the pathogen that causes this disease. So We recently reported this in Plant Disease, so it was a new report. And of course, the big question is, how did this get here? Was it brought from the Eastern US, or was it here already, or did it come from somewhere else? And so one of the advantages when you have these populations already developed, we were basically able to place these Oregon isolates that we collected in this phylogenetic framework. And you can see when you look up here that the Oregon population that we collected, so we have about 8 isolates here, and then we have 4 more isolates down here. They fit neatly into this British Columbia 2 cluster. So it doesn't look like, as our colleagues at the Department of Energy were really worried about, that they brought the pathogen to Eastern Oregon. It looks like this might have moved down from that BC2 population somehow, or been here already where it has caused this disease. So needless to say, there were a lot of sighs of relief when we figured this out. And that takes me to the very last couple of slides in this talk. And that's when I want to talk about the other significant advantage of resequencing these populations, is that you can actually start to predict genes and the function of those genes. So we did the usual thing where we predicted effectors, we predicted CASIs, and then and we found some slight differences that might be interest, but nothing I really want to go into too much detail here. But once we started looking at the secondary metabolite clusters, that's when we found something really interesting. And so we used this anti-smash package to predict secondary metabolite clusters. And we did that for each of these isolates. And as you can see, when we look at these, so each one of these little boxes represents a secondary metabolite cluster. So I have a label here, and I'll tell you what those are in a little bit, but it's these 2 that we were really interested in. And the reason is that basically, especially this 3571 group is basically absent from all of the BC2 individuals, and this 3030 cluster is absent from the majority of the Oregon isolates. So that was really interesting, and very strong correlative evidence that there's something going on here. Then when we group these 2 by their disease severity and their cankers per centimeter, we actually see a similar pattern where we get the individuals who have these clusters present are a lot more aggressive than the individuals who are lacking these clusters. And so here's the predicted, or the annotation for what these 2 clusters do. And then the little really exciting bit about this is that this 3030 cluster you see up here is actually one of the clusters in a secondary metabolite group described in this Dillon paper that is involved in colonization of wood. So it looks like the secondary metabolite cluster that's missing from the least aggressive isolate has also been implicated in the pathogen's ability to colonize wood and cause these cankers. Now, of course, at this point, this is still hand waving and finger pointing. And we're hoping to kind of take this a step further and start to try and do the functional work on this to do knockouts and see if we can get the phenotype to change. So anyways, that's something I'm really excited about in the direction we're kind of moving in. So final take-home messages from this sort of overall look is that we found there's basically 3 genetic clusters in North America. There appears to be gene flow between these clusters, but it's basically the southeastern US and the US-Canada population that seem to be feeding most of the movement of that pathogen. And that makes a lot of sense when you think about where most of the poplar culture and poplar plantations are. There's evidence for extensive sexual recombination within these clusters. And then the BC2 cluster is really interesting to us because of its reduced aggressiveness and the evidence for this lack of these secondary metabolite clusters in that population. So that's pretty much what I wanted to talk to you about today. I hope I was on time and not over too much. And then I just want to present this last slide where basically just to give a reminder of what happens kind of at the landscape level when you have one of these introduced pathogens. So this is southwestern Oregon, Curry County, where you have a Phytophthora ramorum outbreak. And you can see all those gray trees in there are trees that have been killed by this invasive pathogen. So anyways, thank you very much, and I'd be happy to entertain questions.
Thank you, Jared. It was an excellent talk, really interesting to see how this, to see this approach of actually looking at the population of all of these pathogens and being able to track it and link it with metabolites. I found that really fascinating. So we are now ready to answer questions. So if you would like to post a question, please type it into the Q&A box and we will take turns reading them. So I'd like to start with Wenjia Hao, who asks, how was the direction of migration determined?
How is the direction of migration determined? So it would basically, so that if you're referring to the M statistic, it's basically a mutation corrected measure estimate of migration, and you can do it to compare the diversity within the one population to the diversity in the other population, the second population.
Okay, so we don't have any more posted questions right now, so this gives me and Erica a chance to ask ours. But but this is just a reminder that this is your opportunity to interact with the author. You've got the author right here, chance to ask questions. Type your questions into the Q&A box. So not into the chat, but into the Q&A. And but Erica,
yes.
You have anything you wanted to bring up here?
Yeah, of course, it was interesting that you collected from different hosts, right? Host species, populous species, and natural growing trees and plantation hybrids. So why do you think you didn't see any genetic differentiation by host? Do you think that says more about the pathogen or the host?
I think it says more about, in this case, I think it says more about the host. So I didn't go into this in much detail, but the eastern United States has different species that are native to there, so Populus deltoides, whereas Populus trichocarpa is from the western US, and it's a naive host, so it hasn't been exposed to the pathogen. So I think one of the issues might be it's just actually too susceptible, even the resistant ones, to really see these differences.
Okay, well, why don't I read the next question then? We have from Maisa Ciampi Gugliardi, and I'm sorry about that, I'm pretty sure I didn't pronounce that right, who asks, in case of funding restriction, what would be the minimum sample size for population genomics analysis if you already know that you have different clusters? And that's a good question, because that's always a challenge.
Yeah. That's a really good question. I think it would depend a lot on just the genetic diversity that's present in that pathogen. You know, I'm probably answering this in a way that probably everyone knows this. You know, I'm not trying to cop out, but I'm just not sure, without knowing more about the pathogen, what the minimum number would be. I mean, you need to have an idea of the genetic diversity, how much sexual recombination is happening. And you'd also need to, you know, get an idea of how large the genome is. That can make a difference. There's lots of sort of factors that play into this. In a different study with a different ascomycete, to compare populations, we arbitrarily chose 12 individuals as the number we needed to compare different populations. But like I said, I think it varies a lot depending on the actual pathogen or species that you're interested in studying.
Okay, Hyun Fan has the next question. How similar was the genomes of the BC group compared to the other two?
guess I'm not sure how to answer that. What does he mean? Do you mean in terms of like gene content or just diversity, or, I mean, if we look at the BC2 compared to everything else, they're it really is, it's not really that different than all the others. I mean, you can kind of think of this as basically a large population. I'm not remembering the theta values for each of the different populations off the top of my head. It's in the paper, but really they're not that different. There might actually be some other populations in Western Canada, like Alberta, that actually have higher FST values, so are actually potentially more different from each other. So hopefully that answers.
So just to follow up on that, from sort of naively here, you have both the SNP diversity, but you also have this idea of a pan-genome and which genes are there and which genes are not there, which you touched on when you talked about these metabolite clusters. Yeah. So if you looked at it from the point of view of the pan-genome, so does the BC2 isolate have a much smaller genome? Like, is it missing a lot? Is it that it has different genes, or is it just a smaller genome?
It just has different genes. I would say the differences are more, not necessarily in the number, but the content. And so we did some work looking at just nucleotide diversity between these different populations in these predicted genes. And so we see significant differences in nucleotide diversity between these different populations, but there aren't like large pieces of the genome that are missing. You know, there's really the one, like we looked at effectors, we looked at CASIs, and really it was these secondary metabolites that kind of like jumped out at us, which actually didn't even make it into the paper, which is sort of unfortunate, but.
There's always a next, and actually I love it when authors tell us where things are going, you know, things don't just stop at the end of the paper, but what are the connections that you're leading forward. Okay. Do you want to get the next one, Erica?
Okay. Meng Lu asks, was infection rate similar between greenhouse experiment and field?
Okay, so that's a good question. So, I mean, I guess I can sort of interpret what you're asking is if you had the same genotype in the field as you did in the greenhouse, was the disease level similar? So in this specific study, we didn't look at that. We have done other work where we've done correlations between greenhouse inoculations and the field performance. And we see a strong correlation when we look at the extremes. So with resistant individuals, they stay resistant in the greenhouse, and susceptible individuals stay susceptible in the greenhouse and field. So those are good. The middle, we're not as good at predicting. One thing to note here too is that we actually caught this outbreak really early, and kudos to Oak Ridge National Lab, the Department of Energy, and their industrial collaborators, because they told us about this epidemic. We figured out what was going on, and they actually eradicated it. They cut the whole thing down and got rid of it because they were so worried about it. And I don't think that happens very often. And it was like, by the time we figured out what it was basically like 4 months later, it was all cut down. So that made it hard to go back and look at anything else. But I think in terms of a success story for plant protection, I think that was really great.
So do you see less disease in the field where that BC2 cluster is, or do you think those individuals are just so susceptible that they don't need to be as aggressive?
Yeah, I mean, there was a fair amount of disease, but it's one of those things. You have this plantation and this pathogen's moving into it. And so it's like you have all these individuals in there that aren't infected, but we don't know if that's just an escape or if they actually are resistant or what's going on. I mean, so that makes it a little bit tricky. The other thing to kind of add to this is that that plantation where that was happening, it was split in half. One half was irrigated and one half wasn't. And it was the irrigated side that had the infections and not the dry side. So that was kind of interesting too. Pretty hard, but because of that pattern of sort of moving in, it was hard to sort of tease that apart.
So is it always neat like that where you see really just one isolate in these trees in the field? Do do you ever see multiple isolates in one area? I guess I'm wondering whether if they're infected with the BC2, which is milder, what happens then if there is an influx of one of the other isolates?
Yes. So another thing I didn't talk about in the talk was there's two mating types for this fungus. And so in there, we know where they're mapped. And so we actually went in and looked if you had one or both mating types in that Oregon population or the BC2 population. And we actually did have both mating types there. And if you look at that phylogenetic tree, you'll see that there is one clone, so a bunch of isolates that were all the same, but then there's a little group underneath, and they all have longer branches on them. So there's about 4 isolates that were different in there. So I think both things are going on. And we did, like I said, we had both MAT1 and MAT2 idiomorphs in that population.
So this is just a reminder, if you have questions, type them in. Otherwise, Erica and I get to ask our questions. So this is your chance. We'll let Erica's question go next, but really, folks, this is your opportunity.
Yeah, related to the mating and the LD analysis. So if you think of the BC2 as sort of a small population that underwent a bottleneck, do you see similar levels of LD if you look in a smaller population within the other 2 clusters, like if you looked within one particular region, or does it look like it's mixing very well even at the local level?
Yeah, that's a good question. We didn't do that. And actually that would be interesting to add those Oregon isolates to that LD analysis, just because they're more geographically separated, and see if that made a difference. And the other thing is BC2, those individuals were collected from a couple plantations in a small area. And so that could be, I think it's likely an effect of the sampling more than anything else.
So Mike, we have the first author asking a question here. Javier Tabima, the first author. So do you think there can be an opportunity of host specificity between the genetic clusters of spirulina and species or hybrids of poplar?
Yes, I do actually. I think when we start looking at these other, isn't that what I'm supposed to do when he's presenting? Kind of give a leading question like that? Yeah, no, we do think that that's going on. And, the evidence where we see these, I put it in quotations, avirulent type isolates that are colonizing these lenticels might suggest that kind of thing is happening, that this pathogen might, in certain situations, be behaving more like a endophyte or something that's able to colonize the plant tissue. Did I answer that question to your satisfaction, Javier?
He says yes.
Oh, good.
In the chat. Yes, good. Good, you're off the hook. So my question is, what do you think, what is the next big question? I mean, there's so many different directions you could go, right? There's the metabolite clusters, there's, association mapping in populace, there's many different things. So what is the next big question for you in this area?
Yeah, I think the next big question is going after that secondary metabolite cluster. We also have some effector candidates that we're sort of interested in working on as well. But I really think that secondary metabolite cluster and the ability of that group of genes to help wood colonization is pretty interesting and exciting. And we have done a fair bit on the poplar side of things. So we have done GWAS on the host and identified resistance and susceptibility genes there as well. So, I mean, there's, I think, the opportunity to start looking at specific interactions if we can just line them up. They line up, the stars align.
Yeah. I think go ahead.
Oh, I just have a sort of naive question, which is this pathogen found outside North America? I know poplar is attractive to pathogens. Does this help you? If not, does this help you learn about what might be going on in other popular pathogen interactions in terms of mechanisms that it's spreading, that it might be spreading?
Yeah, so it's been introduced to Brazil, and I think Chile as well. And then there's also some places in Eastern Europe, in Russia and the Urals, where it's been described. But we don't have access to any of those isolates, unfortunately, because I think that'd be pretty exciting to be able to like put them into that tree and see, okay, this is where they came from. The other thing that'd be really interested in looking at is there's a couple sister species of spirulina. And doing some comparative genomics work between them, because the sister species are also not able to colonize wood. They just cause a leaf spot. So I think that'd be another interesting avenue to explore.
Really interesting to see which are the genes that control whether it can get into the different kinds of locations that it infects. I had another question, but now I've forgotten it. But fortunately, we have another question written in, which is Hayun Fan asks, in terms of simulation on different levels of LD, how do you do that? Did you filter your SNP data into different groups based on LD, with LD being linkage disequilibrium?
Yeah, so I think if you want to have a look at how that was done, it's probably better to go look at the paper that's referenced in here than to have me kind of try to explain that. So I think my email is available. If you want, I can send you the link to that email, but it's referenced in the paper, and it's actually referenced the lead author on this paper is also the lead author on that paper as well. So I could get you the paper. It's probably the easiest way to answer that question.
And that's a good point for me to remember to say, that whenever you are reading these papers, so I hope that many of you will go back and look at the details in the paper that Jared LeBoldus was talking about today. But remember that the corresponding author always has their email address there. And so that is a way that you can connect with the author and ask your questions and really develop a bit of a dialogue, asking about some of the details, because that's one reason we have, that reminds me to tell you that we've made this paper open access so that anybody can go and check it after the talk. And I hope you will, and look at some of the details. And if you have questions, contact Jared LeBoldus and you can get some more discussion of the details. And Ashley has just posted the link to the paper again in MPMI. And so we've made it open access through the end of the year, but of course, MPMI is going fully open access in January 2021. And so at that point, all of our papers from the previous year will be open access. So we hope that this will draw you in to look at this paper and some of the others. I have one last question for Jared, which is just, I've been thinking a lot lately about this transition to more virulent to less virulent with pathogens. So you have these isolates of the same pathogen, which are more or less virulent, right? The BC2 isolate versus the others. And you also have this closely related species, which is not able to be virulent at all on wood. So I just wondered if you could just comment on this transition that you can see here. And I wonder what some of the pressures might be to which have led BC2 to be less virulent or the other strains to be more virulent.
Right. So that paper, that Dillard paper I referenced, in that paper, they basically talk about horizontal gene transfer from penicillin, I believe, that's able to colonize wood. And so that's their hypothesis for the mechanism of how that pathogen got that ability to colonize wood. And, looking at their paper would be a lot better than me trying to explain it. But, you know, I think that's definitely something that happens maybe a lot more than we realize. You know, and in terms of, yeah, the sister species, and I mean, it's an interesting question. I don't know if I have any more insights really than that. So.
But seeing that whole diversity of isolates that you've looked at really gives us a sense of that spectrum, that it gives us a much more nuanced look of the pathogen and how it interacts.
Yeah. And all the noise that you have to deal with when you're doing experiments with it.
all the noise, yes. Anyway, well, this has been terrific. I don't know if you have any last questions or comments, Erica.
No, I think I've gotten all my questions answered.
And Jared, thank you so much for talking about your recent paper, and for Javier Tabima, for our first author, for jumping in. I hope that people will take a look at the paper for if they want more, to dive more deeply, and that this will give people a chance to think more about a population genomics way, an approach of thinking about getting a much more nuanced view of the interactions between plants and microbes, especially in this pathogen. Jared, any last thoughts or words, suggestions, comments?
No, I just want to thank everyone again for coming today and for the invitation. And thanks again to Ashley for coordinating all this as well. So
yes, thank you. And I hope people will join us in 2 weeks. We have another very interesting seminar coming up. I believe it's Julia Galota, who will be talking about chitin responses, molecular responses to chitin inside Fiscometrella. So yes, long ago, before there were trees, before there were vascular plants, plants had reactions, had responses to chitin that in many ways look very similar to what plants like the flowering plants and gymnosperms, et cetera, have. So that's going to be, we're going to be doing a different kind of evolutionary look, going backwards in evolution and trying to see, get a glimpse into how everything evolved. So I hope you'll be able to join us in 2 weeks. Okay, thanks everyone. Really enjoyed having you join us, and hope to see you in 2 weeks.